remarkable little tuna forest, and last year we got some beautiful imagery, and you can actually see that these tentacles will protrude for meters, and so they kind of are having out their long lines to fish in a water column, and that's absolutely spectacular to see. So yeah, the density has remained fairly moderate on this dive so far. So far, the community seems to be mostly dominated by sponges in the genus Poliopogon and Walteria, these two glass sponges. We just imaged a Walteria a little while ago. The Poliopogon is those kind of larger sponges out in the distance. For corals, we see several chrysogorgid corals, like the one that we just passed here, that plume, that's a Metallogorgia. And then there's several bamboo corals, and then also crinoids. So there's a, if we have time, there's kind of a, looks like an anemone towards the left of the screen, now center left of screen.
Do you want to take a look at this hydroid on the right quickly, or?
Oh yeah, absolutely. So that's a new record for this dive. So that's
a good spot video.
Yeah, well done. So that's going to be in the family Corimorphidae. So that's a hydroid. It's a solitary hydroid, meaning that the entire animal is composed of a single polyp. So cnidarians, the group that contains the corals, anemones, other groups. So you have some individuals that are what we term colonial, meaning that a single animal is composed of hundreds to thousands of these. Oh, beautiful. And that's a synaptobranchic eel just photobombing us. And yeah, so this is right now in our gallery. This is we only have this as Corimorphidae, the genus, but just passing us right there was a synaptobranchic eel, the very first fish we imaged today. Unfortunately, it was in a hurry.
Just pushing him over. So that's one disadvantage of getting closer.
So yeah, this entire animal is composed of a single polyp. So cnidarians, this group that contains the corals, are characterized by having nematocysts or stinging cells. So if you would look at the tentacles under a very powerful microscope, you would find these specialized cells that kind of look like harpoons that they use to capture prey. And within the cnidarian, you have some forms that are colonial, meaning that a single animal is composed of hundreds of thousands of these tiny polyps all joined together living in a single colony, or conversely you have
going a little closer.
solitary forms like this one right here, where the entire animal is composed of a single polyp.
So as we zoom out, I think we have a request from the shore-based science teams to get some lasers so we can get a scale on this guy.
Lasers, lasers on.
Zoom out quickly, Tara. I don't know how much time we have.
We got a little bit.
Just to make sure we got it.
Oh yeah, so that's actually, that puts that into perspective. That's not a small hydroid. It's about a half a meter in height. A little less than half a meter, I would guess.
Steady.
And yeah, so Scott had remarked that this particular hydroid looks very similar to something that we imaged last year. So yeah, we imaged
Okay.
Nice work,
Zoom out.
Tyler.
last year, and then the last couple of dives,
All right, coming up.
Yeah, come out.
Push
several
ahead. Yep.
Yeah.
of these hydroids, and that looks very similar to something that we've.
I'm playing the cautious game.
Yeah, that's fine.
These
I'm
eaves
I'm
are a
all
little
for that.
more than we're used to.
There's a fish. We made that happen. I think.
Yeah, so it seems like there's
Yeah,
a.
well,
That's
see if I can
that's
get
a
it
first.
closer.
There's another one of those Trisopathes black corals.
Looks like a holothurian maybe.
Is that a fish there? That's.
Or a fish now. Fish. Nice.
Okay, quick zoom.
And so I believe
You got
that's
more
that
time
Float along with it.
synaptobranchid eel, I
Oh,
believe.
Oh, nope. Don't want to hit the sponge.
That's okay.
And
Nice.
so, yes, I believe this is going to be Synaptobranchus, either affinis or coplei.
Well, while we're here.
All right.
Nice avoidance technique.
And
No shot was worth hitting. That's
That's right. Well, I know
Looks like
Chris
nice.
Kelly wouldn't be happy. So
so yeah, this is another one of these glass sponges, and this is going to be in the genus Poliopogon, and it's going to be Poliopogon species B, and that's kind of a placeholder for now. We've been differentiating these group of sponges based on how many atrial surfaces. So these kind of vertical surfaces they have. Some have 1, 2, 3, or 4.
Just go up slowly.
And this whole group is distinguished. So if you look at the base, they actually have these hook-like skeletal, modified skeletal components that are kind of hooked and attached to the substrate. And what we're looking at right there is a crinoid to the left, and that's going to be in the genus Clyptometra, and the species Clyptometra lateralis.
okay.
And
So
the
nobody
ophiuroid
push
is going to
ahead
be
again.
a ophiucantid.
Thanks.
Yeah, why don't you try to crab over
Okay.
that center screen?
Sure.
Yeah, ship
So
has
we can get
stopped.
some eyes on this.
Probably
I'll
got
push
maybe
out ahead too.
not even like a minute left of swing, I would guess.
Copy that.
I'll push out ahead until we see another fish, and then we'll get distracted by that again.
Current depth is 1,700
Center up.
meters. So we're just about 100 meters from the summit of the seamount. Again, this is the very, very first time that we're diving on this feature. The very first time anyone has ever surveyed this area. And we were, in fact, successful in collecting both a geological sample early in the dive that's going to help geologists determine the age of this feature based on what we're seeing in terms of the thickness of the manganese crust. I think we're already pretty convinced that this is
Coral on
not
the bottom
Hawaiian,
left,
but that this is Cretaceous.
the black coral.
Yeah.
Yeah, so yeah, that's that brown one to the bottom right. Yeah, that's going to be in the genus Trisopathes.
Do you want to get a look at that, or
No, I think they're
No.
common enough.
okay?
But actually to the left now, the left bottom, there is another, and we have not gotten a close-up of that. Yeah, that kind of looks like a feather coming into view right now, and that's going to be in the genus Bathypathes. And if we have some time, there's going to be
Yeah, I got
at least one very happy on shore-based scientist who would like some close-up of this guy. So I talked before about the black corals or antipatherans, and pathes being a common suffix to a lot of these genera, so they all have that suffix pathes in the back.
Degrees.
So yeah, there's
Get square on it here. Watch lead.
Yeah.
Zoom in a second.
Excellent, yeah, and then potentially where that kind of darker spot is, that towards the top, that's going to be of interest.
Looks like we came over to the more towards the middle of that ridge there. So yeah, 0 to 5 sounds good.
Cool. I think at 40 meters, by the way.
Yeah, let's wait to put that in.
Yep.
So pilot's
Yep.
all set, but that sounds good.
Cool.
Okay, let's zoom in video.
So yeah, we're looking at the branching pattern first. So that gives us an indication of the species. So this is what we've been referring to as comparing to Bathypathis alternata, and then towards the top, there's a dark spot, and that is a symbiotic commensal pollinoid polychaete or scale worm. And that's something that we've started seeing. Are we full zoom? Yeah.
This is full zoom, yep.
Do you need to get closer?
If we have time, yeah.
Go for it.
Okay, let's zoom out video. I'll try to go a little closer here.
Watch lead, is that something that's internal or external?
Well, so there's both. There's some, in this case, that's external, but there are some scale worms that actually are found within the tissues, so kind of nestling in between the skeleton and the tissues. And this is a particularly interesting observation that we've made with this
Sure thing.
this group, is that this polychaete actually causes the black coral to modify its tissues, and it forms kind of like a channel there in the center screen, in the center branch where that polychaete lives. And so yeah, it modifies the tissues completely, and so Tina is going to be very, very happy with that. So yeah, it's an external association. And you can also get a very beautiful view there of the polyps. The top one right there, you see that star-shaped feature. So this is, and this whole family of black corals has a very unique polyps, in that they're elongate transversely. It almost looked like a polyp is composed of 3 distinct segments. And actually, for a long time, people thought that this group was different, and that it had polyps with 2 tentacles instead of the 6 that you have in this group.
All right.
And that's just because the
All right,
polyps are so elongate. So thank you very much for that. But yes, associations like that are very common.
Call that moving.
Polychaetes.
Which direction? Just straight up
There's
the-
about 10,000 species of polychaetes, and a lot of them are actually associated with other organisms living either on top, and in some cases even inside the tissues of organisms.
Are you interested in getting a rockier watch lead?
No, I think we'll push on. Yeah, it's
Okay.
been so common.
Okay.
It looks like you're back to normal. I'd go ahead and call it in.
Cool.
Bridge, this is Nav.
And
Go ahead.
Yeah, I'd like to request a ship move 40
so just to give our shore-based scientists a little bit of heads up. So we have approximately 4 hours remaining on this dive. We'll be pushing off at around 15:30 Hawaii time.
That is correct, thank you.
So we should have a nice long bottom time on this dive. Can we get the lasers, please?
Lasers are on,
Oh,
I believe.
yeah, so they're just in the background.
come down and
They might.
So this is a,
I don't see them.
not horizontal. Oh, there they
yeah.
are.
Yeah, and that actually is that's an enormous unbranched wire coral. So those lasers are only 10 centimeters. My best guess is this is close to 2 meters in height, based on a quick measurement I just did here on the screen. So that wire coral is taller than an adult human being. And again, that's something I like to point out, that a lot of these animals that we're looking at are really enormous animals and that we know virtually nothing about. So, yeah, this is another unbranched bamboo coral. Also a common group that we've seen throughout this dive.
I'm gonna come up and push out a bit.
Copy.
I guess I don't need to push out too far, but I'll come up.
Also want to once again thank all of our onshore scientists that are joining us on this Saturday. We got folks joining us from Hawaii, the ECC, both at the University of Hawaii, as well as the Inouye Regional Center. We got folks from abroad, Tina Molotsowa joining us from the P.P. Shiroshoff Institute of Oceanology, as well as Asako Matsumoto from the Chiba Institute in Japan, and several other scientists. We got Scott France from the University of Louisiana Lafayette, Santiago Herrera, formerly MIT Woods Hole, Les Watling, University of Hawaii, Brian Boston, University of Hawaii, and Abby Lapointe, also University of Hawaii. So yeah, thank you so much for joining us and helping us guide these observations and in participating. We do truly appreciate it.
A little stretched out there.
Okay.
Another unbranched bamboo coral. Also, I'd like to take a second to welcome all of our visitors that might be watching this live feed at one of our public displays that have been set up around the state of Hawaii, including at the Maui Ocean Center, the Waikiki Aquarium, as well as the Mokupapapa Discovery Center. You are watching live video footage being brought to you from the Okeanos Explorer NOAA ship that's spending about 3 weeks up in the northwestern Hawaiian Islands, exploring deep water habitats both in and around the Papahanaumokuakea Marine National Monument. Right now you're looking at a chrysogorgid Gorgonian octocoral. This is going to be in the genus Metallogorgia. The species Metallogorgia melanocristus, one of the more conspicuous species that has that top plume. In most cases, you'll see a little ophiuroid, a Asteroschemata, perching on top of that plume.
Just gonna let it float out of view, but that's okay. Yeah, let's zoom out.
Okay.
Nice.
So yeah, that
Establishing.
it's going to be it's going to be an ophiuroid, or closely related to the brittle stars in the top center, and it's in a different family though. Those are in the Asteroschematidae.
come up as you come up.
So for those folks that are maybe catching this at one of the general viewing displays at several of the aquariums and visitor centers, this video is actually is available over the internet, so you can log on oceanexplorer.noaa.gov, and for the next couple weeks we'll be diving in this area, and so on a daily basis or close to a daily basis, weather permitting, we will be exploring these deep water environments, and anyone can join us over the internet. There's also a mobile app that folks can download, and you can actually watch this video over your smartphone as well. So yeah, there's a sponge there to the top left that's going to be of interest. Yeah, just above the lasers right now. So in the foreground in front of the sponge is another one of those bathypathes, another black coral, but yeah, that we imaged a little earlier, but that sponge is the first time we've seen that on this dive.
Zoom in video. Try to hold it stable.
So we see it's an unusual growth form, but I think it is a polyopogon sponge, just an unusual growth form.
Thank you, Bridge.
And to our right, we see several more of those black corals that we collected earlier, those coiled, unbranched wire corals in the foreground. And actually, if we can catch that black coral in the foreground, well, it's not the best angle, but again, that dark spot in the center is going to be of interest.
Do you want me to move around and try to catch the polychaete?
I don't think we have time for
yeah,
that.
that's all right. Thank you.
Okay. All right, zoom out.
But you might have been able to catch it. There was actually one branch to the top of that black coral where their skeleton was exposed, so the polyps were gone. And you might have noticed that was dark in coloration, or black, and that's what gives this group their common name of black corals, as the color of the skeleton. And yeah, our current depth is 1,680 meters. We're just 80 meters from the summit of the seamount. Fairly consistent, moderate densities of animals throughout this portion of the dive. Also, kind of consistent in terms of the species. Can we maybe snap zoom of that sponge if it's not delayed?
That's a fish.
I'd say
right.
let's get the fish to the right. Yeah.
Okay.
Given that that's only our second fish, I believe it's another synaptobranchid eel.
You've got some
Okay,
time.
zoom
I think
video.
the ship is stopped. Yeah.
Yes, ship is stopped. You have time.
And that's a beautiful view.
Maybe kill the lasers for a second.
Killing lasers. Aye.
No, I was mistaken. That's not a synaptobranchidial. And unfortunately, Bruce, I don't think is joining us, but I'm sure he'll be delighted to see this video. This is something new. We have not imaged this fish before. And yeah, it's not in our gallery. And
Get a full body shot.
so yeah, I think a lot of our ichthyology friends are going to be delighted with this imagery.
Okay, coming in for another close-up.
Hey Scott, can we get
Say that one more time, Alan.
Sorry.
It's fine.
can we get a full front? So hesitate a little bit so we can get the stream.
Excellent, yeah. So yeah, there's a request to just get a view from the head to the back.
From the head to the back?
Yeah, kind of just get a full...
Okay, so zoom in and then pan up.
Correct.
Okay, zoom in video.
This is full wide, or
Okay.
full zoom.
Okay, and then I'll pan up a bit. Let's do the steps here.
Yeah, so no, I don't know this group. I don't know if Alan or anyone else that's joining us. I definitely know that it's not in the gallery of images that we've acquired so far on these two expeditions.
Panning down.
I'm not sure. We need Bruce.
Yeah. But I'm sure he's going to be delighted. And yeah, so last year we imaged just over, well, actually 170 fish during 18 dives. And a quarter of those are completely new records for this area. So most of these fish were only known from samples that were collected very long time ago, and so we're really expanding the amount of known biodiversity of this area, and that's absolutely spectacular imagery there. And
Okay, so zoom out. Do you want me to swim toward him, or do you think we have enough imagery?
Go through, watch this.
I think we're okay, yeah,
Okay.
unless we're parked here, but yeah, I think we're good.
Okay, let's zoom out video.
Cool. It was very
Thank you
flat.
very much. Yeah, good spot there.
Not very spry.
It looks like he's getting pretty old now.
So the ship is not moving, and I'm pretty settled out. So if you want to explore for a bit,
Okay.
that sounds good. I think Nav was just about to say exactly what I just said. Go for it.
Yeah.
So you just said you were hogging another ship.
That was really nice.
Moving.
Thanks, guys.
Thanks, Alan. Yeah, and I'm frantically going through the Hurl gallery of their recorded fishes, and I don't see anything like that. I'm sure Bruce and Ken and many of the others that have been following us will be delighted with that imagery. So current depth is 1,670
Heading
meter
views are. It looks like
meters.
local highs going a little more north than our previous 25 degrees, but what do you think?
Coming up. I think that sounds good. A little more north of where we are now. Like dead north. Is that what you're
Yeah,
thinking? Local high?
now that he's at the top, maybe 25 is all right.
Well, that 025 would make sense on the bathymetry, so maybe we should just keep it there.
Yeah, I like that. Cool.
It looks like it levels out on the left and keeps going up on
Yeah,
the right.
and actually, when
So
came
I
up, you could
just
see a little
updated
bit.
our
Yeah.
position here
Okay.
into Flittermouse.
I'll go 025. You guys ready?
Yeah.
And
Yeah, I think so. You
we
shut it
covered
like
about
fine.
300 meters so far in a dive.
Bridge, this is Nav.
We're still about
Mr. Bridge,
300
go
meters
ahead.
from the summit.
Yeah, I'd like to request a ship move 40 meters, bearing 025 at a speed of 0.2 knots.
That's 40 meters, 025 degrees, 0.2 knots.
That is correct. Thank you.
Coming up.
Yeah. So if we have a little time, maybe that coiled one, coral now to center.
On
If
the
it,
bottom.
yeah, that's
Center
just a
bottom. Okay.
different coloration than the one we collected earlier. And in fact, yeah, there's another in that black coral to the right. So there's
We have
two,
time.
or yeah,
Closer here.
if we have to choose one over the other, the one to the right is going to be a more.
Got a little bit of time.
Let's do a snap zoom on this whip here.
Sure.
Nav bridge, move initiated.
Thank you, Bridge.
It's also going to be a Stichopathes, but now in the brown form. The ones we imaged earlier were yellow-greenish. And coloration is something that's interesting in this group, and that we have several. And so this is a different genus now. This guy with a commensal squat lobster, and that's going to be in the genus Paranthropathes. And good point there, Tina.
Or do you want to get another angle?
Well, are we parked here for a while?
The ship,
Yeah.
I think, is moving, but we've got some time.
Have a little bit of time yet.
Yeah, I mean, basically they want to look at it from like a 90 degrees from the other side, but if that's too complicated, yeah.
I can.
Which way?
90 degrees port or starboard? Around the left or the right?
The left, yeah, port.
Okay. All right. I can try to get it from the water column. And then
No worries if
the
it's
swing
too
arm is
complicated.
okay.
Yeah.
Okay.
Just go.
I'll just come up high enough and look down.
Yeah, we're just looking at the crab, right?
Or the coral. The coral or the crab?
Yeah, so there's a lot of interest in this animal.
Yeah.
So it's not the genus I was
It's not that
originally thinking it was.
Yeah, just put your left toe in, and then control rotation. Steady.
And
Of course, now I've kicked up a bunch of stuff.
It's okay.
Let's try zooming in from here. Let's just try it.
May take your camera.
yeah.
Yeah, sure.
Yeah, so that's exactly what I wanted to see. Yeah, so this is the Hexapathes. That's not the Paranthropathes that I was thinking this is. And so, yeah, it's the first time we've recorded that genus on this expedition, as well as on this particular dive. So, yeah, thank you, Tina, for pointing that out.
We'll get a bit of an establishing.
Got all that snow.
It's kind of pretty.
Yeah, I can stop the ship if this is.
Okay.
That's okay.
I think we're
Good.
Hit
Okay.
it.
we're fine.
About halfway through the move, by the way.
Copy that.
Look at that large crinode.
So now we push.
push?
Yeah.
Okay.
Push so we can
So
stay
yeah,
together.
they're in that large anemone to our right.
Push this
That's
one
a
together.
Venus flytrap or Hormathiid anemone. And then we have a Brisingid sea star to our right there.
Just get a quick zoom from above. Or actually, my camera.
Oh, my apologies.
No problem.
So that's the first time we've imaged a sea star on this dive. And that's
Just a
going
quick
to be...
snap zoom video.
So that's going to be in the family Brisingidae. And then to the bottom, there's a couple crinoids. And
Do
to
you want to get a closer look, or I was about to move uphill, watch lead?
no, that's perfect.
Okay.
Thank you.
Yeah, full wide video. So.
Video's what you got.
So there's a little bit actually a discussion in the chat room, and are we already on a move? That
The ship is moving, yeah.
Yeah, ship is moving. It's
What's the
Yeah.
desire?
about 10 meters left of the move, so a couple more minutes.
okay, yeah. We'll try to keep an eye out for another one of those bottle brush kind of black corals. So that's of interest to potentially collect.
Yeah, well, actually, we're too far past that at this point. Sorry.
Yeah, no worries.
Yeah, I don't know. You just jumped on. Keep Iris up on Zeus Plus a bit. Thanks.
Got a little glare.
So as we keep on ascending up this ridge, I just want to point out some of the landscape that we've been observing almost the entire time on this dive. So we're under the suspicion that the seamount that we're diving on is much older than the surrounding Hawaiian volcanoes. It's actually Cretaceous in age, meaning that it's probably about 80 million years. This volcano has never been dove on and has never been sampled before. We were able to successfully collect a geological
video
sample
contract.
that will be very crucial in
Your live
revealing the
video.
absolute
Thank you.
age of this volcano once geologists can actually get their hands on that and process the sample in laboratories later on. Aside from getting a rock and waiting until we know the absolute age, we can most definitely speculate on the age of this volcano. So if you've seen some of our dives previously, when we were diving on Hawaiian volcanoes, we were able to point out specific types of lava flow morphologies, like sheet flows and pillow lavas, and even flow structures and things like that. But on this dive, you may have noticed that I haven't been able to tell you guys about any of those kinds of features. That's because the surface of the rocks is heavily coated in manganese, concealing any kind of primary features that were placed during those lava flows. Aside from heavy manganese crust suggesting that this volcano is much older than Hawaiian volcanoes, again, getting back to the
Looks like
the
ship is in
scenery that we were seeing on Hawaiian volcanoes, we did see lava flows, and majority of the slope that we've been going up is either completely cemented in manganese crust, or it's this rubbly talus field indicating heavy erosion. So this volcano's been sitting around for tens of millions of years, just kind of breaking apart and slowly falling down into the seafloor. So those two observations paired together definitely support that this volcano is in fact much older than the Hawaiian volcanoes that we've dove on previously.
And combining that, Jonathan, that's very interesting. So we've dove now on a couple of Cretaceous seamounts in this depth range, and the community is, the biological community is distinctive. So suggesting that time might also shape these communities in a very different way. So if you have a few additional million years of extra time, then the communities start looking completely different. So yeah, we did a dive last year on a Cretaceous seamount, and that's just north of here. And yeah, it was also just characterized by these Poliopogon Walteria sponges and a couple of Chrysogorgid corals, and different than other communities that we've seen.
Yep.
Co-pilot's ready?
Pilot's ready. Cool.
Bridge, this is Nav.
This is the bridge. Go ahead.
I'd like to request a ship move 40 meters, bearing 035 at a speed of 0.2 knots.
That's 40 meters, 035 degrees, 0.2.
So yeah, we've been seeing throughout the dive. It's kind of hard to see, but yeah, those wire corals, those coils, very abundant. You know, kind of almost overlook them unless you're kind of zoomed in. But yeah, there you see there's at least half a dozen there in view right now. We have no idea what species that is, but we did collect a sample of that will help us answer that question. To the center of the screen is another Brisingid sea star. They have these very elongate arms.
Nav, bridge,
So yeah,
move
Dan, I'm a little bit curious as to your last statement. So you were saying that you see differences in the biological communities in Cretaceous seamounts versus Hawaiian seamounts. So the time gap that Dan is talking about right now is Cretaceous features being around 80 to 85 million years, Hawaiian features in this area being around 25 to 30 million years old. So a complete difference of 50 million years, allowing for these surfaces to evolve. However, the lifespans of some of these organisms is on the order of 100 to 1,000 years. Can you speculate on any reason why those timescales don't quite agree, or what makes a 50 million year old difference in surfaces more preferable to a certain community?
Yeah, that's a very complex question. Well, yeah, and I should preface what I said before. So it's we have a very limited amount of observations now of diving on Cretaceous seamounts. Just a couple ones that I participated on, the communities look different. We have in ecology the concept of a climax community. So if left undisturbed for a very long time, a community will eventually reach a particular, what they call climax community.
035.
And in areas where there have higher abundance of disturbances over time, you have different species, or more opportunistic species coming in after the disturbance. But then if the disturbances are not happening often, you'll have those opportunistic species, which are more typically faster growing, quick to settle in an area, being replaced by species that are characterized by being slower growing, having lower reproductive rates. And so, yeah, you start seeing different species in an environment if it is left undisturbed over a very, very long time. And that's a concept that's typically, it's been used in terrestrial ecology and in marine ecology. It's still not being applied just because we don't have observations over very, very long time periods like that. We haven't really surveyed many of the marine environments, and especially not deep environments, over large time scales.
Thanks, Dan. So I think that highlights one of the most exciting and rewarding things about studying science is that we're constantly finding new unexplored realms to go and try and see what's there, and as we start to make some observations about some of these biological communities and the geological substrate in which they're growing and living on, more questions arise than answers. And
So has the slope just been pretty steady?
It's been
this
pretty
is
steady
one of the
until
best things about
this-
science is
So I was wondering.
your ability
Everyone
to
said it was pretty gentle.
come up with some sort of creative solution to any questions you might have.
An anomaly.
But you still have the ground 25 meters ahead, so.
Yeah, I've been picking it up consistently in sonar, and then when I come way up, you can see it in the background too.
Good.
And that's how we've been doing our ship moves. I've been coming up, so you can see it out ahead.
Gotcha.
So we have one rock and one bio. Got room for one more of each.
Okay. So this looks like a little local high here. Little pinnacle.
So throughout this dive, we've been talking to you guys about the age of this seamount being Cretaceous, but we're describing modern and live growing biology on this seamount. One really fun thing for a geologist to think about is thinking back in time and what would have been going on when this volcano was actually forming. And so I mentioned that this is a Cretaceous age volcano and about 85 million years old potentially. And so some of the most iconic biology and organisms that were living during the Cretaceous are many different kinds of sharks and bony fishes. Some things that you might have heard of before are some sea lizard type of gigantic organisms like mosasaurs and plesiosaurs, characterized by these large fins that their legs had evolved into, as well as some gigantic clams. They're called rudous clams, about 1 meter in diameter. Many of you might also be familiar with ammonites, and during this period, ammonites started to burst into an evolutionary stage in which they diversified heavily. Ammonites are part of the greater group of cephalopods. Cephalopods roughly translates or directly translates into head feet. On the micro side of things, there were also diatoms starting to make their first appearances during this time. Diatoms are microscopic photosynthetic organisms that are feeding in the oceans. They make their skeletons out of silica. This is a little bit different than some of the other microorganisms that are photosynthetic that preceded them, like Coccolus and Foraminifera. In that, Coccolus and Foraminifera constructed their skeletons out of calcium instead of silica.
It looks like the most recent ship move is.
So when this volcano was erupting on the seafloor, it had prehistoric sharks and fish swimming around it, as well as mosasaurs and plesiosaurs with shallower waters being dominated by rudous clams, a couple of ammonites swimming around and feeding on those, and
yes.
even microbiological organisms making their first appearance.
Sounds 50 sound, is that fine?
Yeah, let's just see what we can find. Okay, once I get past these corals, I'm going to turn to starboard. So we've come off this local spire. I'm turning back in the general direction of the ridge. And you can see, or cannot see, that the ground is not there in the ROV. So once Sirius is over this bump, we'll come down and reacquire the bottom.
Nav, Copilot.
Hey, Copilot.
There we go.
So we're heading 035, right?
That's what we have been.
Okay.
But that ship move just ended about 2 minutes ago.
All right, so we're swinging to a stop?
We should be. Maybe just a couple more minutes of swing.
Okay. pilot nav.
Yes, co-pilot.
My set point right now is a little bit shy of 035.
Yes.
So I'm going to swing right about 4 or 5 degrees.
Copy that. I'll follow you around. I'm out here in this depression between the spire and the rest of the ridge. Just wait until you get over that spire.
Copy. I should be at 3.5 now.
Okay, looks good.
So for those of you who may just be joining us, we're diving on an unnamed seamount that is just southwest of a place called Bank 9, approximately 1,150 miles to the northwest of Honolulu. We're traveling to the northeast up a ridge. Current depth is 1,650 meters. We're trying to make an attempt to gain the summit area of this Cretaceous feature and age. It's approximately 80 million year old volcano that is lying on the seafloor. And once we get to that summit, we're going to continue our survey to the northwest and see what we can find up there. We've been able to successfully collect one geological sample, which will be crucial in determining the age of this volcano. This is the very first time that anybody has ever dove or sampled this seamount, and so that was a monumental achievement by our ROV team to secure our first geological sample, as well as an unknown black coral that they collected about midway up our dive on this ridge currently. So both of those samples will be critical in assessing not only the geological characteristic of this, but the biological characteristics as well.
So the terrain's definitely a little confusing here. Still going up, but I'm basically headed due east here. The ship is about stopped right now.
Yeah, in theory, it should just be out of swing probably about now.
Co-pilot, pilot.
Go ahead, pilot.
So looks like the ridge takes a little bend here. As you can see, I'm facing east and uphill, and I'm off to your starboard. If you could just kind of turn to face me more. The ship is stopping, so we'll just turn and follow the local terrain. We don't have to stick exactly to 035.
Copy that. Right now.
We've had a couple of requests from our shore-based scientists to get a zoom on that coral to the left bottom of the screen right now, please.
Copy that. This one just coming in the left-hand side?
Correct.
Okay. Video, you with me?
Yeah, I'm here, pilot.
Okay. Let's see. I'll just try a floating zoom here.
Roger, going in.
Kind of up in the air. Yeah, just
bring your camera up a bit.
Yeah, go in as you can. Okay, coming up.
Going in. Holding.
Okay, coming out more.
Holding.
Okay. There we go.
There you go. Coming in.
Not a lot of polyps on this. I'll work up to these polyps.
Yeah, they seem to be in the center, higher portion.
There we go.
So Dan has stepped out to lunch. But I'm going to attempt my best at a biological identification. I think what we're looking at is a bamboo coral, and you can tell that it's a bamboo coral by these light and dark alternating bands within the skeletal system itself.
Coming in a little bit more, that's full zoom.
So we're trying to get a close-up for some of our onshore shore-based scientists so they can get a better idea of what we're looking at here.
Feels like we're drifting away.
Yeah, I think I am going slowly back.
So one of the things that we're trying to image is the arrangement of polyps on these branches, and it seemed like that a critical thing to be able to characterize this species is, say, the arrangement of 3 polyps around one of the branches.
I'll try to get the lasers on it here.
Scott France, one of our shore-based scientists, has provided an interesting observation that it appears to have been victim to predation. Possibly one of the reasons that it's lacking polyps on the most exterior branches.
And we imaged one of these a while ago, and yeah, that's what we've been referring to as Isidella species lyrate. Yeah, sorry, I just stepped aside a little bit here to have lunch and just chat about upcoming days. So just to give our shore-based scientists a little bit of a heads up. So yeah, again, for today's dive, we're going to be coming off the bottom in just about 3 hours and 15 minutes from now. Now, weather does not look... oh, there's a huge eel there in front of us. Did we image that already? Whoa.
Well, looks like we're pretty stable here. I'll get this fish, and then we can figure out
that
which
is
way to go.
awesome.
Co-pilot, can you get lasers on? Okay, video, you're free to zoom.
Yeah,
Thank you coming in. I'll hold the whole fish for a while.
Okay, there they are.
So now that's going to be a synaptobranchid eel.
A little tighter. You might be frame right though.
Yeah, Nav and co-pilot, as I do this, if you guys can try to figure out which way we should proceed.
And I'm pretty certain that that's going to be the species Breva dorsalis, or short dorsal fin.
Real quick.
It looks like the dorsal fin is...
Yeah, I'm wondering if your sonar is picking up that pinnacle we just passed.
extends only over a short portion of the body, so that species Synaptobranchus brevidorsalis. The dorsal fin is about 50%, covers about 50% of the body.
Watch lead any particular part of this fish you want? We've got some good overall images.
Yeah, just about maybe on the center of the fish. Just zoom in.
Get him right now.
Okay.
Coming in.
On the top and the dorsal.
So nav.
Yeah, so exactly. Yeah, perfect. That's exactly what I wanted. Yeah. So there we got a good view of that. The dorsal fin. Depending on where that starts on the back of the body, that's a diagnostic feature to differentiate species. And this particular one, brevidorsalis, or short dorsal fin, it starts
I'll move over towards its head.
About halfway on the body.
So I was wondering, maybe we should just
and
going towards.
And yeah, that gives you a nice appreciation of like how common these wired corals are in these environments.
At least for a little bit.
Holding.
We did collect one earlier in the dive, and that's going to be the what's going to help us identify the species.
I come in a little more and show that lateral line.
Yeah, beautiful image of this synaptobranchid eel. One of the more common and conspicuous of the fish members of this community. You see a lot of these eels and rat tails
physically? or
Those two
emotionally?
groups are kind of, and then maybe a halosaurus in addition to that. So those three groups comprise the
Coming
vast
closer.
majority of the fish that we see at these deeper depths.
Okay, Nav, so I'm up a little bit higher off the terrain. And still
So,
don't see this picture.
Yeah, just want to once again thank all of our shore-based